Good morning to everyone. Thank you for joining me in this session. Today, we will discuss about the whole genome analysis of a bacteria. So, as you know very well, bacteria are prokaryotes. So, today we will discuss about one bacteria which I used for my research purpose. So, in bottom left, you will be seeing the micrograph of that bacteria. It is called Verminephrobacter iceni. So on the left side, you see the microscopic image, and the right side, you see the SEM image, that is electron micrograph image. So before entering to the workshop, if you are having your laptop ready, and if you want to do all the analysis along with me, you can use this link and register for this trial of this software. You can download it, and it will be around 500 MB. So it will take some time to download. So if you are able to open it in your laptop, then you can visualize all the data sets in your system itself. I will just paste this link in the chat box. Okay. Start with, so in the left side, if you see, this is called Icenia fetida. It is also called the red wiggler earthworm. Our lab was mainly focusing on the genome sequence of earthworms. And this Icenia fetida had its genome sequenced by some foreign scholars, and it was already available in the website. What we did was, we just took the genome sequence and we started working on it. We looked for some biological features inside the genome. As you know, genome is nothing but a single copy of the entire DNA present in an organism. It is called a genome. Genome is like a genetic blueprint of an organism. Just like you have a blueprint for a building, you have the genome sequence. It has the information about all the proteins which are coded in the genome, all the RNA which are coded in the genome. So if you have the sequence, which is nothing but the alternative arrangements of random arrangement of A, T, G, and C. If you have the sequence and if you are able to analyze it and get a meaningful data from it, you have the whole blueprint of the entire organism. That means you can map all the morphological features to the DNA sequence. So if you want to analyze the sequence of an organism, first you have to take the organism, extract out the DNA or isolate the DNA from the tissue, and then you have to go for sequencing. Sequencing is nothing but finding out the order of arrangement of ATGC nucleotide bases. So, as you very well know about Sanger dideoxy methods, now recent methods called next generation sequencing have very sophisticated instruments which do the automatic process of Sanger's dideoxy method of sequencing. So you have various platforms like Illumina, Iron Torrent, Pacific Biosciences, like that. So each of the technology will use each of its own process to sequence the DNA piece. Each of them have their own limitations. That's from there you can get the DNA sequence. Then you can analyze it to get meaningful information. Before we go to bioinformatic analysis, it is very good to have a little idea about the biology behind it. Because without biology, bioinformatics data will be very dry. Only the biology data makes it more interesting. So if you see this earthworm, it will be having many segmented, it is a segmented organism. It is just like a train with many number of compartments. So in the second image, bottom image, if you see, there are many compartments, and in each compartment, you will be having an organ called nephridia. This organ is equivalent to our kidney. Since this is a lower organism, it is not a fully developed kidney, but it will perform the function of a kidney. It is having a 3-loop-like structure, and through one pore, the fluid in the earthworm will enter inside, get filtered, and the waste materials will be sent out to the other opening. Now, if you see, these earthworms are having a particular type of bacteria living in their nephridia. So Those type of bacteria are named Verminephrobacter. Vermi means worm. Nephrobacter means the bacteria living in the nephridia of the worm. So these earthworms are called farmer's friend. They improve the fertility of soil. They live in the soil. And in the soil, you find a vast variety of microbial population. So these earthworms will be having many bacteria inside their body and on the surface of their bodies. But specifically, this Vermibacter is observed in the nephridia of this earthworm. This, as you know, these earthworms are hermaphrodites. They possess both male and female sex organs in the same worm. But two different earthworms act as parents and they produce offsprings. They actually lay eggs, which are technically called cocoons, which you see in the right side diagram. So these cocoons will be having tiny worms inside them. Once they hatch, small worms will come out and grow into full size. These offsprings does not have direct contact with the parent worm, but still they will be having Verminephrobacter in the nephridium of the offspring. So scientists were very eager to know how it is happening. So how it is happening means, if you see diagram B, there is a fully grown worm and this portion called clitellum will pinch off from the worm. While doing so, it will take away some bacteria from the body surface of the parent worm. And this cocoon which you see is a fluid-filled body and it will be having a protein called albumin. Albumin is a rich protein source. So these bacteria thrive in the albumin of this cocoon. Once the cocoon is released out, slowly the embryo will develop. And while the embryo is developing, if you see diagram C, there is a small gray color pore here and a red color marking. These are the bacteria. They slowly enter the canal by means of type 4 pili. You would have learned in microbiology about pili and flagella. Using type 4 pili, they attach specifically to the canal. It is called a recruitment canal and in the embryo, and it directly goes to the nephridia. Once it enters inside, there are 3 loops. The middle loop is called the ampulla. So slowly it moves with the help of flagella to the ampulla area, and there it colonizes. So as the worm matures, the nephridia will start functioning. Once it starts functioning, it will filter out or purify the coelomic fluid of the earthworm, and it will excrete the nitrogen waste. These bacteria will utilize that nitrogen waste and they thrive in the ampulla. So thereby, this earthworm is providing shelter and also food source to the bacteria. So this is called symbiosis, whereby both of the organisms will get benefited. It is already known that this earthworm genome does not have a gene for producing vitamin B2. But vitamin B2 is essential for an organism. So this earthworm will take vitamin B2 from other bacteria. For example, this earthworm will be feeding on dead and decaying matter along with soil. So the bacteria in the soil, they will enter the gut of this earthworm and they will start to produce vitamin B2 and secrete it out. This earthworm can absorb that vitamin and it can use it for its purpose. That is well known. But this Verminephrobacter, it is specifically colonizing the nephridia, and it is passed from one generation to other generation in a well-specified manner. So we got a doubt that is it living there only for vitamin B2 or is it going to give some other benefits to the earthworm? So what we did was we were just analyzing the genome of this earthworm, and interestingly what we found was this genome of this earthworm was containing a genome of bacteria. Actually, scientists were isolating earthworm. Since it is living in the soil, they used to clean it very well and then extract the tissue and extract the DNA. But by accident or without knowing it, they actually took this nephridia tissue. And inside this nephridia Verminephrobacter was present. So while sequencing, they sequenced both the earthworm genome and the bacteria genome. So if you see this diagram, the blue color one are the earthworm DNA and the red color one are the bacterial DNA. So when they published it, they thought the entire thing as a single organism. But when I analyzed it, we came to know that this red color bars indicating the bacteria genome were also present in the data set. So what I did was, I took the genome of the type strain. Type strain is something, when you first name a bacteria, you deposit all the information of the bacteria in a database. Now that strain, in this case, EF012 becomes the type strain. This bacteria genome is represented as a closed circle because it is a, it is a circular chromosome. It will be of the size 5.5 megabase pairs. That means 5.5 into 10 power 6 number of bases. A normal E. coli will also having 5 megabase pairs of DNA. And also the number of genes is around 5,000. That is the normal number of genes for a bacteria. For a human, it will be around 3.2 into 10 power 9 bases, and number of genes will be 20,000 unique genes. So here this is a bacteria. So we had the type strain reference genome. Using that, we separated all the reads of the bacteria and we assembled back into the Verminephrobacter Icenia MSU strain of size 4.4 megabase pairs. Actually, we got only 4.4 out of 5.5. There is a loss of 1.1 MB, but this is good for a bacterial draft genome. If the genome is complete, we call it as a complete genome. But if a genome has some DNA pieces lost, we call it as a draft genome. Now, coming to the sequencing part, I told you for sequencing a bacteria, you need to culture the bacteria, then isolate the DNA, and then sequence the DNA. But in this case, I found the sequence hidden in the earthworm genome. So luckily, I don't have to do the culturing and isolating bacteria again. I took the reads. Now for sequencing, this sequencing technology have some limitations. Like for example, this is a 5.5 MB of single piece DNA, but you cannot give it as an input to a sequencing machine as such, because it cannot take 5.5 MB size of DNA. It can take only DNA of small pieces. So what scientists do is they just take the DNA, they randomly cut it into small pieces, and those pieces they put it into an input of a sequencing machine. And then the sequencing machine generates an output. And that output too will be of a specific size. For example, if it is a short read sequencer, it can produce sequences in the range of few hundred bases. If it is a long read sequences, it can produce sequence in the range of few kilobases, 1,000 bases long. So in this case, the sequences were from a short read sequencer, which are around the length of 100 base per long. So these are the steps of a sequencing workflow, where you take the DNA, you fragment the DNA, then you add some adapters, because you don't know which pieces are fragmented. It is a random one. So you add small pieces of DNA to it, and that small pieces are called adapters. Then you create a library, and that DNA library is amplified by using a PCR technique. Then you put it inside the sequencing machine, and then you get an output. That output is called a sequencing read, READ read. These reads vary in size for different sequencing. For example, if it is an Illumina machine, it will produce a short read sequence of length around 100 bases long. Now, you imagine a situation. You imagine this whole genome of a round circle is a Hindu newspaper for today's newspaper. You imagine there will be many bacteria. So you will have many copies of this chromosome. So you imagine the newspaper for a month, for the month of July, and you have uh, multiple copies of the same newspaper, say 100. So there will be 3,000 newspapers. Now imagine it for 1 year long. You will be having many newspapers. What we are doing is we are just tearing down the newspaper. It will be shredded into small pieces. Now, if I ask you to assemble it into a single newspaper, is it possible? Do you think, is it an easy task to assemble back all the papers which you tore down? It will be a very difficult task. But in this case, what is happening is, as you see here, there will be overlap between two different reads. Because these two reads come from different genomic pieces, from different bacteria. So they are torn down in random way. So there will be overlap between them. Using that, we can get a consensus sequence. And from there, we can produce the genome again. This process is called an assembly. But even doing so, there will be some gaps in the genome. You cannot get a complete circle, but there will be some gaps. It is called a draft genome. So these are the steps followed in a sequencing workflow for a bacteria. First, you get the reads. Before going to assembly, you check the quality of the bases, because the machine gives the output, but it will not be 100% confident. It will give a confidence value. So based on that, we will check the quality of the reads, whether it is okay, or if it is below the threshold score, we will be trimming them out. We will trim the ends of the reads so that we get only the confident reads, confident portion of the reads, and we go for assembly. There are many tools to check the quality. One typical example is FastQC. This is the name of a software which is used for quality check. Second process is called assembly, wherein you take the reads, you align them together just like a multiple sequence alignment, and then you take a consensus. This will give you an assembly of a draft. Now, once you get the chromosome, you have many genes in it. So one gene will be taking a span of around 1,000 bases. So in last session, you would have tried ORF, Open Reading Frame Finder. These are similar programs which can be used to find out the genes in the chromosome. One famous software is called Augustus, which you can use to find the genes in the DNA or the entire chromosome. Once you find the genes, you don't know the name of the gene or the identity of the gene. You just know this is a gene segment, but you don't know its name. There are many ways to find out the identity of the sequence. One way is by using BLAST. You have a DNA sequence, you want to find the protein name. So you do a BLASTX. Or if the protein is not there, or the homologous sequence of the protein is not already there in the database, you can go for a protein domain search by using InterProScan. Then you can name it. Since in our case, this bacteria is already having a type strain, it will be having all the names by using BLASTX. By using BLASTX, we can derive all the names of all the sequences. Then you can identify the pathways encoded by the genome, encoded in the genome by using KEGG annotation. KEGG stands for Kyoto Encyclopedia of Genes and Genomes. This is a database wherein it has information of all the genes involved in all pathways. By comparing your genes with the database, you can come to know the metabolic ability of the bacteria, whether it can degrade an protein, or does it have any antibiotic resistance. All these details you can get. Then comes the non-coding genes. These are some specific RNA like microRNA or 16S RNA, ribosomal RNA, tRNA. All these details you can find by comparing it with the RFAM database. Infernal is a tool which you can use to do the comparing job for you. So this is the quality checking reads, and this is the typical result of a quality check. So in the x-axis of this diagram, you see the position of the reads, and in y-axis, you see the quality scores. If the quality scores is above 20, it is okay. And, the regions are color coded. If it comes in this orange region, it is okay. If it comes on the green region, it is better. So in this data set, if you see, all the reads are coming in the green region. So it is quality wise, this is okay. So the next step is assembly. As I told you, you can use Augustus, Glimmer, or any such softwares for assembly. Once you assemble it, you will be having the reads will be converted into contiguous sequences. So once you convert into contigs, you get your draft genome. And this is called a summary table, where you can summarize all the information about the assembly. So if you see here, the total number of reads were 0.5 million. Number of clean reads, a little less than that. We use FastQC, then trim the unknown thing, and then we keep only the confident reads. The average length of clean reads was 89. Since I told you, this is a Illumina short read sequencing output. So you have reads of average length 100 bases only. So here the average length was 89 after trimming. And you have the percentage for that. And you have the assembly contigs. So there is a total of 1832 contigs. In your material folder, you will be having the reads, and in assembly folder, you will be having the contigs file. So in the material folder, if you go inside step 0 reads, there you can see the read_1.fastq, read_2.fastq. These are nothing but your reads. It is named fastq because it is just a FASTA file along with quality scores. So FASTA plus quality becomes FASTQ. So these are the reads file which you can take as input. And in step 1, you have the assembly. In assembly result, you are having a file called contigs.fasta. If you open that, you can see this 1832 number of contigs. Now the maximum contig length is 31 KB. And the minimum contig length is 500. And the average length is 2.4 KB. As I told you, one gene will be in terms of 1000 base pairs. So the mean contig length is 2.4, which is well above 1000. So this assembly is a good one. If the sequencing is of poor quality, this mean contig length will become very less. So in those type of contigs or contiguous sequences, you cannot find any meaningful genes. So those are not a valid assembly, but this one here is a valid assembly. Then you have N75, N50 values. This N50 is a value in statistical terms. So you can express the quality of the assembly in statistical terms. What you will be doing is, you are having 1832 contigs there, of different length. The maximum length is 31,000 and the minimum length is 500. You arrange all the contigs in descending order, starting from 31,000 and ending in the 500 base pairs length contigs. Now, look at the exact middle. The exact middle of the 1800 sequence will be around 900 position. So in position 900, you see a contig and you take the length of the contig, and that length is 3593. So this is called the N50 value. So this N50 value, if it is higher, it is better. If it is very small, that means the assembly is poor because it is not having sufficient length contigs, so that the N50 value, the length of the contig in the middle is very less. So the length of the contig in the middle, if it is somewhat bigger, you can say that this assembly is a good one. Then comes GC percentage. GC percentage is nothing but the percentage of guanine and cytosine bases among the 4 ATGC bases. This GC percentage parameter is an important one. This GC percentage can be used as a parameter to distinguish the type of the organism. If you take a bacteria, this GC percentage will come above 50, normally in the range of 60 to 70. Here it is 65.5%. So if you take some other strain of Verminephrobacter, it will be having a GC percentage close to 65. If you take this earthworm, in which this worm is residing, that earthworm will be having a GC percentage of around 40% because it is an eukaryote. Eukaryotic organisms will have a different GC percentage. That means the composition of GC in their genome will be very less. So by looking at the GC percentage itself, you can identify whether the genome is a eukaryotic origin or a prokaryotic origin. Now, the size of the genome is, it is having 4.4 million bases. Now, on the left side, you are seeing an image. This is called a genome map. We are having a genome of size 4.4 megabase pairs. It is not possible to show you all the 4.4 bases in a single slide. So to make it simplified, we can represent it in a diagram by means of a map. So this is in the form of a circle because your chromosome is a closed circle. So in the outer ring, you are having some arrow mark, and the inner ring also you are having arrow mark. These two arrow marks will go in opposite directions because you know a DNA will be having two ends, the 5' end and the 3' end. So these bacteria or eukaryotes will have genes in both the directions. A gene can be present in the 5' to 3' direction and also in 3' to 5' direction because this DNA is double-stranded and each strand can code for the genes. So this diagram depicts the genes or the distribution of the genes in the genome. So next comes the prediction of the genes. You have the entire chromosome, DNA. Among the DNA, only specific parts code for proteins. That is called a gene. So you use different softwares to predict the genes. A simple technique would be to use ORF finder. ORF means open reading frame. The region of DNA coming between one start codon and one stop codon is called a ORF, open reading frame. So if you are manually taking the DNA sequence and putting in an open reading frame software or a finder, and if you're taking the result, that will consume a lot of time. Because here the number of contigs is 1832. You cannot do it 2000 times manually. So we have softwares which do the function automatically. These are called gene prediction software or gene finding software. In ORF finder, the logic is you look for start codon, which is AUG, and you look for stop codon. And it will report the region between these two codons. But in addition to that, these softwares, for example, Augustus, will take into account the already existing gene models. You would have sequenced a bacteria already, and you know the gene features of the bacteria. So you create that as the model, and using that model, you predict the genes in the new bacteria. So if you look at this diagram, this is a gene diagram from a eukaryote, not a bacteria. For your understanding, I have kept this diagram here. The black line is the genome, and in the genome, this gene is present in 3' to 5' direction, that is reverse direction. So on the right side, you are seeing a start codon, and before that you have an enhancer sequence, then Tata box, then the green region is the gene of 5810 bases. From the gene, the red color line indicates the mRNA or the complementary DNA. Then from the mRNA, you get the CDS regions, the yellow color arrow marks. And in between the yellow color arrow marks are the non-coding regions or nothing but the introns, the intervening regions. These yellow color arrows are called exons or the protein expressing regions. Expressing regions is combined and you get the word exon. Then this is in the case of an eukaryote. But in the case of bacteria, you don't have exon intron. You have only exons. Then this is the protein. Now, this is representing the gene structure as a diagram. So we need a way to represent it as a digital form to communicate with the computer program. So there is a format called GFF, generic feature file format. So I have given a screenshot of the format here. Just like your FASTA format is to store DNA sequence. Similarly, GFF3 format is to store the annotations. Annotation is nothing but labeling the DNA sequence. For example, if you see in the top diagram, this green color region is labeled as gene. So this is called an annotation. So in the FASTA file, you will be having a header and the sequence. Header will be the first line starting with a greater than symbol. So if this gene is protein disulfide isomerase, you just in the header file, you add protein disulfide isomerase. That is called labeling. In bioinformatics term, that is called annotation. Now, once you annotate it, you need to represent it. So you have a 9-column format. This is called GFF3, generic feature file format version 3. So in the first column, you will be having the contig ID. Here we are having 1832 contigs. So we give the contig number. For example, contig 1. And in the second column, we give the name of the software which is used to predict the sequence, predict the gene. So in this case, it is Augustus. In the third column, you have the name of the feature, whether it is a gene, whether it is a mRNA, whether it is a coding sequence, start codon, stop codon, data box, or some other thing. Then in the fourth column, you see the start of the number of days. For example, this gene is starting at 678 days and ending at 6,480. That will be the fifth column. Then the sixth column, you will be having the score of the prediction software. Then in the seventh column, any additional information. And the in the seventh column, you will be having the direction of the orientation of gene. Here it is a minus sign because it is running from the 3' direction to 5' direction. If it is in the forward direction, you will be having a plus sign. So in the seventh column, you will be having the frame of the feature. For example, every 3 codons code for one protein. So this you are having 3 frames. If it is the first one, starting from the first one, it is called 0. Second base is 1. Third base is 2. That is coded here, 0, 1, and 2. So by this, you can check whether a feature, for example, a coding segment is in frame with the reading frame. Now the 9th column is the extra information. You can give any information. For example, any mutation in the feature, you can type it here. Or any special sequence name also, you can give it in the 9th column. So this is called a GFF3 file. This file can be passed by any bioinformatics software and they can produce a diagram which you see in the top. You cannot port this diagram anywhere, but you can, this text format is highly portable. Now, once you predict the genes, next step is annotation, that is finding a name for the gene sequence. If the sequence or the homologous sequence is already available in the NCBI GenBank database, you can use BlastX to find or retrieve the annotations. If something is an unknown one, you can use interpro. You can scan for the protein domains present in the sequence and you can name the gene. And then you can look at KEGG database to find out the metabolic pathway details. Next comes the non-coding RNA part. So non-coding RNA means, for example, you would have drawn phylogenetic tree by 16S rRNA sequences. That 16S ribosomal RNA is a gene which codes for an RNA which will be part of the ribosome. It will not code for a protein. Similarly, you have many number of non-coding genes in the bacterial genome. You can use the infernal tool, and the database will be RFARM database, and you can pull out all those details. So since here you are having only the genome, you have to look for the 16S rRNA gene. So the best way is you can use an infernal tool and you can predict this is the gene region and just take only that gene, and then you can go for BLAST to identify your bacteria if you don't know the identity of your bacteria also. Then in our case, we are having a Verminephrobacter genome sequence from the earthworm gene, but already there is a type strain. Somebody had already isolated the bacteria, sequenced it, and it is already deposited in NCBI. I was interested in knowing whether it is the same strain as the already existing Verminephrobacter. The only way to do that is I can pull out the 16S rRNA from this genome and compare it with the 16S rRNA of already existing Verminephrobacter ICM. So by the phylogenetic tree, I will come to know whether it is the same bacteria or a different one. Actually, this was a different one. So we gave a different strain name as MSU. After analyzing all these things, we came to the conclusion. We performed all the annotations and then the metabolic pathways. And finally, what we found was these are the vitamins which are present in the bacterial genome, which will be helpful to the earthworm, because earthworm is not having genes for producing all these vitamins. This bacteria will be giving all these vitamins to the earthworm. Out of all these vitamins, it was proven that vitamin B2 gene was not present in the earthworm. In addition, this bacteria is having all these genes. It will contribute the molecules to the earthworm. This was our finding. Now, if you have installed the omics box software, we can proceed with the analysis. Hope you have installed the software. If you have successfully installed the software and you are able to open it, just type a yes here in the chat box so that I will be able to know it. For analyzing a bacterial genome, you can do it in free resources of free software also. But for your understanding, if you are using multiple softwares, you will get confused as to which software to look for. So I am using this omics box, wherein you have all the softwares in the same area, so that you won't be upstream. But this is a commercial software you have to pay and use. But for all these tools, you have the alternative open source version. For example, FastQC is freely available in the link which I have given in the PPT. But the same thing, you can use it here. The only thing is, here the number of genes is more. So the result or the output file which you get from a bioinformatics tool, you have to use either R programming or shell programming or a Python to organize the results. But these commercial softwares will organize the result for you. So your time will be saved. That is the only difference. But all the tools you can use outside also. So this is the FastQC software download, where you can download and use the FastQC. Now you don't need to download FastQC. You can use it in omics box itself. But this is the link. You can go and download here. Now I will show you how to perform FastQC on the read spy. You have the read spy in the step 0 reads folder. There you will be having read underscore 01 and read underscore 2 dot fastq. What the 1 and 2 signifies is in next generation sequencing, to improve the countability of the sequencing, what they do is they sequence in the 5' to 3' direction of the DNA piece, and simultaneously they sequence in the reverse direction 3' to 5'. So the result from the 5' to 3' sequencing will be your forward reads, and the sequencing from 3' to 5' of the same piece will be your reverse reads. So for a particular piece of DNA, you will be having two sequencing results, one forward read and one reverse read. These are called paired reads because they will be complementary to each other. So these two forward and reverse reads are separated in two different files, and they are named _1 and _2.
நம்ம கேள்விப்பட்ட விஷயம் நம்ம பேசணும் அந்த டைம்ல நமக்கு ஒரு சார் எக்ஸாமினர் வந்தாரு நாளைக்கு ஒரு மேடமோ நான் லீவு நல்ல வேலை லீவு
Now here you are having many options. So whatever analysis you are doing, you can save it in .box file or .b2g file. You are having one more variant of omics box software. It is called Blast2Go B2G. So that is an older version of this software, and it is available free to use. But it is not having all these features, but it is free to download and use. So you can directly open the B2G files. So for all the analysis, I have given you .box files. You can also, if you are not able to complete the analysis, you can directly open the .box files. Then you have load option to load different data. So if you see the bottom menu, you are having general tools, genome analysis, genetic variation, transcriptomics, functional analysis, and metagenomics. And you have predefined workflows also. We will be using this general tools, genome analysis, and functional analysis. So if you see in general tools, first you are having faster tools and then fastq tools. Inside fastq tools, you have short read quality assessment with fastqs. You have to select that. And then here add files, you have to choose the reads. And then click next. Here you can provide any adapter sequences which you know. Every individual sequencing company will be having their own adapters. If you already know the adapter sequence, you can give the sequence of the adapter region. Otherwise, this software will try to find it. So I'm clicking the run button. So it will start to run. Yeah, it is finished. And here you will be having the report. You can save it in PDF form. In right side top, you will be having the save as PDF option. You can click it and you can save the entire thing as PDF. This will be your result. If you want to see the individual diagram, which I showed in my PowerPoint, you can go to this table. Here you have the fastq files. Then sequence quality is checked in the first column, sequence quality scores, and all these things. And finally, adapter content and overrepresented sequences. What we can do is we can look for this report for forward. And here in results section, you have per base sequence quality. Click on the chart. So this chart will open. So if you see here, the quality score is on the y-axis, and all the reads are having quality scores above 20. Above 10, 20 to 30, it is good. If it is 30 to 40, it's better. So some of the reads are having their quality in this orange region, 20 to 30. So in that case, you can go for trimming. If you go to general tools, fast queue tools, second option, you can process the reads by using trimming. So what trimming will do is, it will trim the low quality ends of the reads. The sequencing machines, when they give an output, only in the end, they make some mistakes. So or in the end, you may find the adapters. So if you want to clean the adapters, you can just use this Trimomatic software. It will look for the quality scores and trim at the both end of the reads. You don't need to manually open the sequence and delete it. It will automatically trim it. This Trimomatic is also available in open source. You can download and use it. And while downloading and using the softwares, your system, you may be using a laptop or a PC. Your system should be having enough amount of RAM. RAM is the primary memory. For example, for these type of tools, since this is a bacteria sequence and it is less than 5 MB, you can use a normal laptop. But when you go for a human sequence, which is in a GB size, 10⁻⁹ bases, you may need a high RAM computer. If you want to check the RAM of your computer, you can go to this PC or your My Computer, and then right click on there and go to properties. So here, if you see in system, under system, your processor and your installed memory. Processors is nothing but your CPU chip. There will be variants, like if it is Intel, you will be having i3, i5 or i7. For performing genomic analysis, a processor which is i5 or i7 is good. i5 is good, but less than i5 cannot withstand this large amount of data. This is actually a big data once you complete all the analysis. So you need to get a powerful system. Then second one is the installed memory or random access memory, the primary memory. This primary memory, if it is 2 GB, then that laptop is not good for this analysis. If you run multiple softwares like this, your laptop will be in problem. So if you have a RAM of around 4 GB, it is good. If it is 8 GB, it is very good for genomic analysis and opening and visualizing genomic sequences. So you have to check the system in which you are working for a smooth operation of your software tools. So this is all about your quality checking. If you want, you can save the reports. I have already performed this analysis and it is available in the step 0 reads result folder. You can directly go to file and open file .box and choose that box file, .box file. These results will open for you directly if you are not able to perform the analysis. One interesting thing about this software is this Omics box, this company is offering a cloud server. That means, for example, if you are performing an assembly, that assembly program will not run in your laptop. The data will be sent to the server of the company, and there it will be run and the results will be sent back to your computer. So that even if your computer is having low RAM, you can just import the data, do the analysis in the cloud server, and you will get the result. So next step is to do genome assembly. For doing assembly, you go to genome analysis. And there you select DNA-seq de novo assembly. There is two types of assembly. One is de novo assembly and another one is reference-based assembly. Reference-based assembly means you already have the genome of Verminephrobacter. You are sequencing a different strain of Verminephrobacter and using the already existing one as reference. By comparing that, you can arrange the reads and get the conditions. This is called reference-based assembly. If you want to ignore that, because that will be a kind of bias, or if the reference genome is not available, you are sequencing an algorithm for the first time, then you can go for de novo assembly without the reference gene. You directly check for overlap between the reads and then get the consensus conditions. So here in this software, you are having Abyss, Spades, and Fly. These are some algorithms which you can use for assembly, de novo genome assembly. But some of the algorithms will not work for paired end reads. In our case, we are having paired end reads. So we better go for this first option, Abyss. We click next and we choose the read files by using this add files function. And then we click on next. These are some parameters which you have to check for your alignment. For example, one of the thing is minimum alignment length. We know the typical length of a genome is 1,000 bases. Because a protein to be a functional one, it will be having around 300 amino acids. So one amino acid will be having a codon of length 3. So 300 into 3 will be around 900. So a particular gene will be having 1,000 bases. That is our calculation. So if you are aligning very, very small pieces, that is useless. So we can keep that the minimum alignment should be around 40. This is a default one. If you want to improve the quality, you can increase the number. Or if you want more coverage, you can just decrease the number based upon your requirement. It's better to keep the default value. And then click on next button. Then you have to choose where to save the results. For example, here it will be saved as conduct.fasta in the same folder. And then you click run. Once you click run, in this software, what it does is it will send the data to a different server, as you can see here. So it will take around 20 minutes for sending the data and getting it back. If you are using a normal software offline, then you can get the data there itself. There are some softwares which do the assembly program in a fast manner. They use some heuristic techniques, but those data are not reliable. You may miss out some of the genes when you go for such kind of assembly. So this will produce a result which I have given in step 1 assembly, context.fasta. That will be the output. And just like your quality control step, you will be having your report generated in PDF format. If you are not able to complete this step, you can directly use the context.fasta for the next step. You have any queries in this, you can just share me in the chat box.
Sir, excuse me, sir.
Yeah.
Sir, Omexbox is a trial version only, right, sir?
Yeah.
Okay. Is there any other tools, sir, available, sir?
For this workshop, you can use Omexbox.
Okay. Okay. Yes, sir.
Is there any problem getting the trial version?
No. I got the key. While registering, I'm asking it's a trial version for 7 days only. That's why I'm asking.
It's a trial version. It's a commercial software. My point is, we discussed about FastQC.
Okay.
You can individually download FastQC and install it and use it. It is free.
Okay.
Then Augustus, you can individually go and download Augustus and then use it. But for this session, we cannot spend time on downloading all the softwares.
Okay.
So I'm suggesting you take a trial version of this commercial software, get trained, and then you go back and use the individual software separately.
Okay, sir. Okay, fine.
What I'm doing is I'm just aborting this function because it will take 20 minutes of time. I have already stored the result in assembly folder. From there, you can get the context file. I will just open it for you. So this is the context file. I'm opening it using normal WordPad. This is in FASTA format. So this is the contig_1, the first contig. Similarly, you will be having 1832 contigs here. So this is called FASTA format because it is having a heading line followed by a sequence line. So in this heading line, we are having the information just like contig_1. We know this is a gene sequence, but we do not know what is the name of this gene. So our next step is annotation to find out what is the identity of the sequence, and we will be just writing it over here. If it is protein disulfide isomerase, next to this contig_1, we will just write protein disulfide isomerase. Since we are having thousands of sequences, we cannot do it manually. So we will allow the software to do it. So the software will take this FASTA file, just write the name of the sequence here in the first line next to contig_1, and then it will give it back. Now this sequence is called an annotated sequence. So I will show you how to do it. So first you can go for BLAST_X. BLAST_X, you can directly do it from omics box also. So you go to functional analysis. So before that, I have to find the genes to arrive at this contigs. You have the contigs. First, you have to find the gene sequences because ORFs will be embedded or hidden inside the contigs. So what I'm doing is I'm going to genome analysis and then gene finding. So here it is asking whether it is eukaryotic gene finding or prokaryotic gene finding. In prokaryotic gene finding, as I explained before, there will not be any introns. That means intervening regions, the junk DNA. So the region between one gene will be having one ORF. But in case of eukaryotic genes, one gene will be having multiple number of ORFs and multiple coding sequences. So in our case today, it is just Verminephrobacter. So I'm giving a prokaryotic gene finding, and it is asked for the assembly file, that is contigs.fasta file. So I just choose the contigs.fasta file. And choose next. So here it will ask what is the genetic code. If it is bacteria, we will choose the bacterial code. If it is mitochondria or a protozoan, the code may differ. Then it will ask what is the minimum gene length to look for. By default, it is set as 110. You can increase it up to 200. Then next. Then these are some other advanced options. Then I just click run. Again, this software is called Glimmer. Glimmer is a software similar to Augustus, but it is very basic than Augustus. So it will send the data to its server and it will give the results back. An alternative to this Glimmer is Augustus. I will explain you why Augustus is better. So this is the job submission page for Augustus. Here you can manually take some sequences and paste them. Our contig is of size 4.4 megabases long. So you cannot paste that much huge amount of data in this text box. So one way is you can download this Augustus and run locally in your laptop, or part by part, maybe a 5 contigs together, you can paste it and get the result. So once you paste it here, it will ask organism. You are having eukaryotes as well as fungi and bacteria. So here for bacteria, you are having 3 organisms: E. coli, Staph aureus, and another one bacteria. So if your bacteria is gram negative, you can choose E. coli, or Staphylococcus aureus the other way. you don't have a Verminephrobacter here, but you are having an E. coli, meaning this tool has the gene models of E. coli. So when you use this tool for E. coli, it will predict more accurately than Glingman, than randomly selecting or looking for ORF, because you will be having many pseudogenes in the genome. You will be having one start codon and stop codon and a region between, but inside this bacteria in real time, no protein will come out of it or no RNA will come out of it because that gene will be silenced. So those kind of genes are called pseudogenes. It may look to you like a gene, but it is not coding for any protein as such. So you can choose the bacteria and then you can upload any evidence here or that, then just submit it. Then you will be having the result. So just for your reference, I will try to submit one sequence. Asking report genes on both strands or only the forward and reverse strands. It is asking for alternative transcript also. I'm selecting none because this is a bacteria. Splicing is not here. Just click Augustus. So this will be your output. It is predicting the gene. First two lines will be the information about the software, and then there is your GFF file format, the 9-table format which I explained to you. First will be the country ID, then the software, then features, start, stop, and other information, followed by the gene sequence. Then the protein sequence or the amino acid sequence of the protein. Similarly, this is called a GFF3 format and it's giving you the result. Now, if you are doing it manually like this for all the 1,000 countries, it will take months long time. But if you are using downloading software offline and doing it, you will get it. If you are using Omics box, the results will be organized and it will be given to you. So now what I'm doing is it will take long time, maybe more than half an hour. So I'm just aborting it. You will find the results in the results folder. Step 2, gene finding. This genes.fasta is the result, the number of genes present in the genome. So for this run, it will be around 3,800 genes were predicted. And then you have the GFF file. You may try to open the GFF file in WordPad and visualize the gene structures in text form. Now I am just opening the genes.fasta in Omics box just to give you a so here I can visualize the contigs individually. Now we are seeing contig 1. We can choose different contigs and you can see the sequencing. We proceed to the next step, which is annotation. So for doing annotation, you can come to this functional analysis bar, where you can load the sequences. Directly, I can open the box file, open file. So here the contigs and the details are presented as a table and the results are loaded. For example, the first annotation which you can do is blast. For doing a blast, you can just copy the sequence . For example, I'm just copying a sequence. Here you will be having many variants of blast. Since we are having nucleotide and a protein, I'm just choosing blastics. So here I'm just pasting the sequence, giving a job title. Yeah. I want to the database is in a NT database. Just blasting. And then you are having the algorithm parameters, where you will be having the E value. Here it is set it as 0.05. Let it be the default value. And just paste. If you want to do it automatically using omics box, you can go to functional analysis after choosing all the sequences. Here you have blast results. Just give blast. It will ask different options. You can give NCBI blast online. If you have NCBI blast plus software downloaded in your PC, you can do NCBI blast in local option. Now we can do it in online. Give next. Here it will ask for your email ID. You can choose the E value and the database here. And then you can choose the word size. By default, it is 6. If you want to speed up the process, you can decrease it. Or if you want to get a more accurate result, you can increase the word size. Choose a folder to save the results. Actually, the blast result can be downloaded in many formats like HTML, TXT, XML, or XML2. So you have to choose the format which is compatible with the downstream software tools. Once you click run, these results will be sent to blast and the results will be taken back. And you will be having a label as it is blasted. So this is the blast result which I have done manually. So if you see the result is showing the organism is Verminephrobacter icini, and you have the sequence. So we don't need to download it individually. You can go to download all, and in that you have many options: JSON format, XML format, or text format. it's better to download as multiple file XML2 form. Once you choose it, your result will be downloaded. This file, you can manually load it in Blast omics box of, and you get the annotations like this. So you get the identity of the sequence. For example, this contic1.g1, it is a FADR family protein, and then it is having a length of 738. You are having only one hit for that, the E value for that, the similarity percentage, and the number of GOs. GOs is nothing but gene ontology terms. For example, I am working with a protein called superoxide dismutase SOD. I am giving a name to the protein as SOD, superoxide dismutase. Somebody working in a different country on the same protein, he may not be aware that I have already named the protein. He will be giving the name as free radical scavenging protein, FSP. But FSP and SOD are the same protein, but they are given different names. So this will lead to heavy confusion, because I will report in my article as SOD and he will be reporting it as FSP. Then the readers may get confused whether FSD and SOD are two different proteins, or are they talking about the same protein. So to avoid the confusions, there are different gene ontologies. This gene ontology process has been invented. In this, you will be having 4 different processes, and each process will be having its own GO terms and one GO ID. For example, this protein is able to bind with the cell membrane. That is a GO term, ability to bind with the cell membrane. If it is binding with the DNA binding will be an ability, and it is a GO term, DNA binding. That GO term will be associated with the protein. So one protein will be having many number of GO terms based upon its domain and the functions. So it is better to map the GO terms. We have a BLAST result. We are having a homologous sequence. Homologous sequence means 2 sequences which share a common evolutionary origin are said to be homologous sequences. So if that protein is having a particular GO term, that GO term will also be applicable to our protein of interest, because they are having sequence similarity of 100% or above 90%. So the GO terms will be transferred to our sequences. Once the GO terms are transferred by using an algorithm, it will be matched for validation. Once it is validated, it will be given as mapped. That step is called mapping in omics box. Once mapping is finished, you will be having the GO terms associated with your protein in particular region. So once you have the GO terms, you will come to know the ability of the protein, or in which biological process this protein is getting involved. So using that GO terms, you can go for further annotation. If your protein is not there in the database, then it will be labeled as unknown. That is, no researchers or scientists have yet researched on the protein or worked on the protein. In that case, you cannot leave it as such. You have to give it a name. So you can go for looking in the Interpro databases. This Interpro database will be having data of already used studied protein domains. So domain is nothing but a particular region on the protein which is, or which can fold independently so that it is involved in a particular function. So we will check for the presence of any protein domains in this sequence. Here also, same way you will be having a text box where you can paste the protein sequence and search for the domains. Here also the same problem, you get the result, but you have to do any R programming coding or Python coding to combine all the results and give the final data. But in case of this omics box software, it is automatic. So similarly, you can download the results in XML format. I have given you some results in annotation folder. If you are loading the sequences, you can go to that Interpro XML file and few result file I have already given you. If you have these sequences, what you can do is you can go to functional analysis, Interpro scan, or you can go to load Interpro scan results. Either you can do Interpro scan from Omics box itself, or you can separately do the Interpro scan, collect the results in XML format. That is very important because there are more than 11 different formats to download the result. You have to specifically download it in XML format. And once you come back here, you can just click load Interpro scan results, add to existing project, and you can select all the files. And then you have to click load. Once you click load, it will be automatically loaded. It will be annotated based on the Interpro ID. Using this Interpro ID, you can get the domain. For example, you are having an unknown protein and it is having a DNA binding domain. You can change the name of the protein to DNA binding protein. Instead of calling it as unknown, you can call it as DNA binding protein for your further analysis. And the last thing is you are having the geoterms and all the annotations. The final thing will be to make meaning out of it. You can go for KEGG analysis, that is Kyoto Encyclopedia of Genes and Genomes. This is a database maintained by Japanese, wherein they maintain all the details of all biochemical pathways. Here you can browse about all the pathways present in the bacteria or a virus or humans. Depending on the organisms, you can browse, or depending on the genes, you can browse, or even you can browse based upon the diseases. It will have all the biochemistry pathways. Now, we are having a list of genes, annotated genes. Now we can see what are the abilities of the bacteria, what proteins it can produce, or what enzyme it can produce. Thereby, it is able to metabolize which are the things, or whether it is synthesizing a particular vitamin or not. So you can manually go to KK database, KK tool, and submit your sequences and get the results. Or here you can directly go to functional analysis and do combined pathway analysis. So these things will be taken into the database and you will get the result. So this is how we came to know that this Verminephrobacter was able to synthesize all these vitamins: vitamin B2, thiamine, biotin, propionate, pyruvate, folate, and pantothenol. And we did the same for the earthworm genome also. In that, we found that vitamin B2 was not present. Then we were so sure that earthworm was not able to produce vitamin B2, but it is getting vitamin B2 from Verminephrobacter. So in this symbiosis, this bacteria is producing vitamins and giving to the earthworm. Some of the researchers, what they did was, they took the cocoon and they introduced antibiotics to kill this bacteria. And then they observed the mature earthworm. What is happening to the worm? When they did that, what they found was those worms were not maturing in the right time. They took long time to come to sexual maturity, so that they did not produce enough offspring. So this worm was helping them to reach the sexual maturity in a faster pace so that it can reproduce faster or it can become mature faster rate. So the farmers may get more number of earthworms for their feed. So this is about annotation. You can download this or save this thing by using export, and you can export it as a Excel file and store it in your system for further work. Then the final thing is finding out the non-coding genes in the genome. Non-coding genes are nothing but the genes which produce RNA, but that RNA will not be translated to a protein. For example, the ribosomal RNA. The well-known ribosomal RNA is 16S rRNA because you use it to find the phylogenetic relationship of the bacteria with other bacteria. So now we are having the entire whole genome. From the genome, we have to check or get only the 16S rRNA. So for this, the process is same as finding a protein coding gene, but the tool will be different. So you can use the RFARM database. Here you will be having the details of all the RNAs, bacteria, human, and everything. So what we will do is we will use a tool to compare this database with the data, our one. So in Omics box, you can do that, or you are having a tool called Inferno. You can download the Infernal software from here, from edilab.org, and then download the RFARM database, and in your PC you can run by you giving the condigs as the input. So I will show you do it from Omics box. Go to functional analysis and then RFARM RNA family search. Choose this option. And here it will ask for query sequence. You just give input the condigs file. And click run. So the sequences will be sent to a server and you will get a result. Similarly, this GFF files, this non-coding RNA features can also be saved as a GFF file. So this is all about the basic annotation of a, or the analysis and annotation of a bacteria gene. Now we will discuss about the applications of this genome sequence. You are having a bacteria, you isolated the genome, sequenced it, then by using this bioinformatics tools, you have analysis. You are having the annotated genome. What can you do with it? That depends on the biological problem you are working with. In my case, I was working with this earthworm and the bacteria. Verminephrobacter not only lives in this earthworm, it is living outside the earthworm also. So what I can do is, this earthworm and the bacteria are having a close relationship that we know by the name symbiosis. So what we were thinking is this bacteria is constantly supplying some vitamins to the earthworm. Then earthworm is taking it. We had a doubt that where does the gene for that vitamins come from? Are they the genes from earthworm or from the bacteria? So to answer that question, you can go for some advanced level of bioinformatics analysis called pan-genome analysis. Pan-genome analysis means you have a bacteria and the bacteria belongs to a genus. For example, this Verminephrobacter Icenia belongs to Verminephrobacter genus. And another genus, you have many different species. Icenia is one of the species of Verminephrobacter. Now, these Verminephrobacter will be having a gene pool. Some of the species may having entire of the genes, some of them may be having the 95% of the genes. Some may be omitted in some species, but the entire gene set is called a pan-genome of Verminephrobacter. Now, we can look for whether this genome of this our Verminephrobacter Icenia is accepting any gene from other Verminephrobacter. Or even if we consider the earthworm and bacteria, we can find out whether this genome is accepting some other DNA from the earthworm also. Because you know about bacterial transformation. Transformation is nothing but accepting naked form of DNA from the environment. So these bacterial cells, if they are transformation compatible, they can take up some DNA. This earthworm may shred some of the DNA in the outside its body. And there is a lot of chance this bacteria can take the DNA from the environment and integrate in its genome. Now, this bacteria will produce the gene product from that integrated part, and it can produce some vitamins and give it back to the earthworm. In turn, the earthworm may give food and shelter in its nephridia. So this is one kind of work where you can see the openness of the genome, the readiness of the genome to accept from the extra DNA. When scientists study the entire bacteria, some genomes are called closed and some genomes are called open. If they are open genome, currently they are accepting DNA from other sister species or other symbiotic organisms. So that is one kind of analysis which can be done. The other thing is, if you are thinking this symbiotic relationship in a commercial way, you want to commercialize it. For example, this earthworm is a farmer's friend. It will improve the soil fertility of the farming fields. You want to introduce this there. Your Verminephrobacter will be going along with the Icenia foetida earthworm and it will contribute to its sexual maturity and the growth of the earthworm. So that field should be free of any antibiotic contamination which is going to harm Verminephrobacter. If you see here, Verminephrobacter Icenia is very intelligent. It is not inhabiting the first loop of the thing. It enters through the nephrostome and it goes to the second loop. It is inhabiting a safe place whereby it is not in ready contact with the soil. But if it is inhabiting near the pore, easily it can come in contact with antibiotics. But even then, if there is some contamination of antibiotic in the soil, which can harm the population of this Verminephrobacter, then that may damage the beneficial effect of this Icenia foetida. So we can predict that by looking at the gene. How means there are many antibiotics. There are antibiotic resistance genes and antibiotic sensitivity genes. We can look for any antibiotic sensitivity genes present in the genome of the bacteria. And then we can predict that if this antibiotic is present, this population is vulnerable. So we can avoid that. Or if some resistance is not there, a resistance is there, we can predict that this is resistance to this antibiotic. So we can do these kind of efforts. To enlarge your field of knowledge, I will give you an application example. A few years back, there was an outbreak of disease in the goat population. That goat is used for mutton biryani, and you can enjoy mutton biryani. One goat may cost for around 5,000 rupees. So if this bacterial disease comes to the goat, it immediately dies within few weeks. So 5,000 rupees is loss. But what that team members did was they isolated the bacteria. Now they know the bacteria by means of 16x sequence, but they don't know a way to kill the bacteria. They may use different plating techniques, but that will consume a lot of time. So immediately they went for genome sequence. They sequenced the genome, annotated the genes, and then they compared with different databases. And by comparing with different databases, they came to know that they are having different antibiotic resistance genes. That bacteria was having a number of antibiotic resistances. But at the same time, it was susceptible to few antibiotics, few common antibiotics like chloramphenicol, fenicol. So what they did was they formulated a set of antibiotics for which that bacteria was vulnerable. It was a simple bacillus. So what they did was they gave a combination of 3 antibiotics. They tested in the lab and then they gave it to the goat along with this food. The cost of antibiotic treatment for 3 weeks for a particular goat is just 25 rupees. And the antibiotic worked perfectly because we know we have evidence from the genome analysis that that bacteria is susceptible to the particular antibiotic. So it cured the disease in 3 weeks. So 5,000 worth of goat is saved by 3 antibiotics worth of 25 rupees. With 25 rupees, a 5,000 rupees goat was saved and farmers were very happy. So this is one application where you use the knowledge from genome analysis to come up with economically important ideas. The other thing is, if you look in this project, I was able to find a bacterial sequence inside the genome of a eukaryote, that is the earthworm. Now there are reports that viruses are hiding inside the genome of bigger organisms. Like, take for example, human. A human is getting a viral infection. The virus will enter inside the cells and it will deposit its DNA inside the genome. Then the DNA multiplies. The viral DNA will become dormant, but still the sequence is present inside the human gene. So if you want to, being a student, if you want to start a small project, what you can do is these genomic DNA are available free of cost in NCBI. You can go there and download it using the GenBank accession number. If you think this particular Yersinia fetida genomic reads, the sequencing was done way back in 2015. At that time, it would have cost around 20 lakhs to do this. But then the researchers freely deposit it in a database. From there, you students can easily download the sequences. If you have the knowledge of these bioinformatics tools, you can use it to predict the sequences or annotate the sequences present in the genome. You may come up with discoveries like different viruses present in the genome. As I told you, this RFAM database, it has different RNAs of viral origins. Like different RNA. These RNA, you can get a result in your RFAM analysis. I think this is finished. Any results here? Okay, no problem. So once you do this kind of analysis, you will get a prediction as to some viral DNA or viral RNA is hidden in the genome sequence. So you can, by the knowledge of these softwares, you can do your own work with the use of your PC itself. Then you can have a small idea about a very important discovery about the role of that virus in this eukaryotic genome. You can use these tools to initiate a project like that. So I think I have covered the thing. So do you have any questions? Or do you have any doubts which I have to repeat again? Yeah, thank you.